So, good morning, dear students. So today we are going to start with the next part of the sexual reproduction in flowering plant. So today we are going to start with the... Okay. Yes. So, technical glitch. Now, the last topic that we have talked about was about megasporangium. If you remember, the structure of ovule. So we're going to start from there only, and then we are going to move into the next topic. Okay? And mind you, my dear students, this is the topic where you need to go line by line of this chapter, NCERT. Okay? And also the understanding is required. So too much of information from outside is not that requirement, but this is a very important chapter for you, especially for NEET examinations, okay, and definitely for board exams if you are studying for the board exam this year. So let us start with the topic which is called as megasporangium. So when we talk about megasporangium, so normally we know that this is the gynoecium, which is having stigma over here. This is stigma, this is style, and this is ovary, Inside ovary, what is present? Inside ovary is present that particular structure which we call it as the megasporangium or ovule. So this is the structure which we call it as placenta. Fine, because this stalk, which is called as funicle, joins with the ovary. That particular contact is called placenta. Now, so from here we have got the idea that this particular ovule is present inside the ovary. Now, let us find out the structure of this ovule. If you remember, last day we talked about that this is the structure which is called as the embryo sac. This is a structure which we call it as a embryo sac. And inside this embryo sac, you can see there are 3 cells over here, which we call it as antipodal cells. Fine. Now, over here, there are 2 nucleus which we call it as N and N. We call it as polar nuclei. Or secondary nucleus. There is a large egg cell having n number of chromosomes. This is the egg cell having n number of chromosomes, and these two are the synergids. Okay, so this is the particular embryo sac. Now mind you, my dear students, inside this embryo sac, we call, what is the other term for embryo sac? Female gametophyte. Very good. So the other term for embryo sac is female gametophyte. मतलब क्या है? The structure which holds a female gamete. What is a female gamete over here? Egg cell. So gametophyte, phyte means plant body, and the structure which holds the plant body is called gametophyte. The structure which holds the gamete is called gametophyte. So female gametophyte. The other term for female gametophyte is embryo sac. Now, outside this particular structure, what you can see is the first inner integument. This is inner integument. Then, again from here, you can see another structure going up called outer integument. And then this structure coming like this. Now this structure is now called as the funicle, the stalk, and the place where the funicle joins with this outer integument. There is a scar in the seed. We call it as hilum. This is hilum. Okay. Now, Outside this embryo sac, what you can see is there are some group of cells which provides nourishment to the growing embryo sac. And what are these? These are nucellus. These are nucellus. These are nucellus which provides nourishment to the growing embryo sac. This particular opening, you can see, this particular opening is called micropyle. Micropyle. And you can see there's some structure which comes out from the synergids, and this structure which guides the entry of the pollen tube inside the synergid is called filiform apparatus. It's called filiform apparatus. It's called filiform apparatus. Okay. Just opposite to this micropyle, what is present over here is chalaza. What is present over here is chalaza. Chalaza. Okay? Now this is the structure of a typical anatropous ovule. This is the structure of a typical anatropous ovule. Now see, earlier when the stand of NEET was quite good enough, that is during our times, we had to face the questions different types of ovule like ampylotropous, amphitropous, hemitropous, orthotropous. These are not there right now in your course. Fine. So this is anatropous ovule, which is the most important one and is being given in your book. Now, this is the structure of ovule, which we also called as megasporangium. Megasporangium. Now, today's topic starts from here. Today's topic starts from megasporangium and embryo sac. So the pistil, megasporangium, that is ovule, and embryo sac. Now, what it says? What it says. Each pistil has three parts. What is that? Stigma, style, and ovary. So stigma, style, and ovary together makes a pistil. So this is one pistil. Okay. Inside the ovary is the ovarian cavity. Inside the ovary there is a ovarian cavity which is called as locule. Which we call it as locule. Okay. The placenta is located inside the ovarian cavity. The placenta is present over here. Just now I have shown you this. This is the placenta inside the ovarian cavity. What is written over here? The stalk of the flower is called pedicel. We know that the stalk of the flower is called pedicel. So pedicel is this one. This is the thalamus. This is the pedicel. This is the pedicel. And this is the stigma, style, and ovary rest on the thalamus. If it is superior ovary. Okay. Now stalk of the leaf is called petiole. So if this is leaf, this stalk is called what? Petiole. Tell me if you remember, in Mimosa pudica, that is in tuck-me-out plant, the petiole gets swollen. And the petiole gets swollen. And what is that swollen petiole called? Good. The swollen petiole is called as pulvinus. The swollen petiole is called as pulvinus. So stalk of the stamen is called filament. Stalk of the ovule is called funicle. Okay? So this is very, very important. Next is what is about the structure which is being given in your book. You can see the same structure is given in your book. Now, last day we have talked about when many of the androecium they join together, we call them as what? When many of the carpel they join together, we call them syncarpus. We call them syncarpus. So this syncarpus is in the case of what? This syncarpus, see, this is the syncarpus structure, and which is present in multicarpillary. Apocarpus is different. Apocarpus is what? Where the apocarpus is the carpels are free. Apocarpus means free carpels. So they are not fused. And syn means what? Fused carpels. Over here they are the fused carpels. Fine. Now see in the B1. So multicarpillary syncarpus pistil of papaver. So this is papaver somniferum. Papaver somniferum. It reminds you about something, I think, if you remember it, in the topic human health and disease.
Drugs.
As a drug. What kind of drug is that?
The coke.
Coca alkaloid. Coca alkaloid?
Opioids.
Opioids. Power somniferum. Now this is in case of hibiscus. You have seen this is 1, 2, 3, 4, 5. So in hibiscus there are 5 carpels. So we call it pentacarpellary. This is style. And inside the ovary, if we cut the section of it, slice of it, a section of it, what we can see inside, there are 5 chambers, and we call it locules. So if we cut the section of this particular ovary, we can see there are 5 chambers, 1, 2, 3, 4, 5, and inside this, you can see ovules are present. So we can call this as pentalocular and pentacarpellary, pentalocular. And what is the placentation? Axial placentation, because they have got chambers. Axial placentation. So what is placentation? It is the arrangement of the ovules inside the ovary. And there are 5 different types of placentations we have talked about in the chapter morphology of flowering plant. And those are number 1, basal, marginal, axial, free, central, and parietal. Good. Now, this is what? Syncarpus in case of avar. This is apocarpus. And apocarpus in case of Michelia oblonga. Michelia oblonga. Fine. And then this is the diagram of the typical anatropous ovule. Fine. See, this is micropyle. Opposite to micropyle is what? Chalaza. So these are over here. What is present? Synergids. This is the egg cell. These are 2 polar nuclei, and these are 3 antipodal cells present over here in the chazal end. Just now we have drawn this. Fine. This is hilum. Okay, this is the stalk, which is called as a finicle, micropylar pole, outer integument, inner integument, nucellus, embryo sac. So what we know again, let's get into that particular topic. What we know about this particular ovule, what happens to the ovule after fertilization? What happens to this ovule after fertilization? This particular ovary after fertilization becomes what? Fruit. And we call this as a true fruit. We call them to be true fruit. What happens to this ovule after fertilization? It becomes seed. So ovule changes into seed. What happens to this? If this is seed, a seed is having outer covering and inner covering. This is an outer hard covering, we call it as testa. This testa is nothing but the modification of outer integument. Outer integument after fertilization. Because the ovule is going to turn into seeds after fertilization. So what is going to happen? The outer integument changes into testa. And this inner one is called, inner covering is called tegment. So tegment is nothing but, it is a modification of inner integument. It is the modification of inner integument. Is this clear? So this is the particular structure of the seed. You can see an opening over here. This opening is called as micropyle. This opening is called micropyle. And there is a scar over here. This scar is called hilum. Okay. Now, let's come back. See, arising from the placenta are the megasporangium. Arising from the placenta are the megasporangia. Megasporangia, Gia means many of them. Commonly called ovules. The number of ovules in an ovary may be 1, that is in case of wheat, paddy, mango, so they have got only single ovule. And in case of papaya, watermelon and orchid. So it becomes star point for your NEET examination. So they're going to ask you, the ovules present in ovary, if there is only one of them, they are present in wheat, paddy and mango. Papaya, watermelon and orchids, they have got many of the ovules inside the ovary. Now, this structure you can see is nothing but the development of the female gametophyte. development of the female gametophyte. I'm just showing you, it's very, very important for board exam as well as for NEET. What happens? Before this cell, you can see, before this cell, there is a structure which we call it as a archisporial cell. Before this cell, we can see a structure which is called as archisporial cell. Archi means primitive, archisporial cell. And from archisporial cell, this is the structure which comes up called megaspore. So you can see this structure has become megaspore mother cell. And there are some cells present over here which is called nucellus. You can see this is nucellus. Okay, this is the micropylar end, this is the chalazal end. This megaspore mother cell is having a large nucleus. And megaspore mother cell is having 2n number of chromosomes. So what it is going to show first? Mitosis or meiosis? It's going to show, megaspore mother cell is going to show meiosis. It's going to show meiosis 1. And it results into 2 cells, 1, 2, each having n and n number of chromosomes, and we call them as megaspore dyad. Megaspore dyad. This megaspore dyad, which is again as having the nourishment providing cells called nucellus. And this megaspore dyad, it shows what? Meiosis 2, which is called mitosis. Which is called mitosis. So it forms into 2 cells and 2 cells together, 4 cells called megaspore tetrad. Megaspore tetrad. So each one of them, they have got n, n, n and n. But out of these 4, 3 of them degenerates. And what remains is one megaspore, functional megaspore. Functional megaspore. Functional megaspore. But mind you, functional megaspore is this cell. Functional megaspore is this cell, having only one nucleus. This is functional megaspore. And from this functional megaspore, there is a first free nuclear division. What is the meaning of free nuclear division? Nucleus divides, but without cytokinesis. Without cytokinesis. So only karyokinesis is going to occur without cytokinesis. We call it free nuclear division. And it's a free nuclear division 1 which takes place over here. This is called free nuclear division, FND 1. Results into 2 nucleus formation. Again, FND2, free nuclear division 2. From this nucleus, you can see 2 nucleus being formed. And from this nucleus, 2 nucleus being formed without cytoplasm. That means now one cell is containing 4 nucleus. One cell is containing 4 nucleus. And there is a free nuclear division number 3, resulting into 8 nucleus. So one cell, 8 nucleated stage. But now, what is going to happen? In this one cell stage having 8 nucleated stage, some of the cytoplasm will be taken up by 3 types of cells. We call them as 3 types of cells we call them as antipodal cells. 2 nucleus bearing 1 cell, that means it is a 1 cell having 2 nucleus. So it is a 1 cell, 2, 3, 4 antipodal cells, 2 synergids, 4, 5, 6 and 1 egg, that is 7. So now it has become 7 cell stage but 8 nucleated stage. 7 cells, 8 nucleated stage. This is very, very important. This is how the development of female gametophyte occurs. Now, only 1 meiosis takes place and 3 mitosis takes place. 1 meiosis and 3 mitosis takes place to give rise to what? A female gametophyte. Now, this is the antipodal cells. There are 3 of them. They have taken some of the cytoplasm and cell wall and they have made antipodal cells. These 2 together makes 1 cell called central cell, and see, you can see the 1 egg cell is there and 2 synergids present over here. So 1, 2, 3, 4, 5, 6, 7. So 7 cells but 8 nucleated stage. This is the most important thing about what? About this development of female gametophyte. Okay. If you are going to sit for a board exam, then development of female gametophyte can come as 5 marks question. Development of female gametophyte, you have to draw this. Along with that, you have to draw archisporial cell. The megasporangium. the megasporangium, that is ovule. What it says? What it says? The ovule is a small structure attached to the placenta by means of a stalk called funicle. 2011 question. The body of the ovule fuses with the funicle in the region called hilum. So funicle joins with the body. What is that body? Outer integument. Thus, hilum represents the junction between ovule and funicle. It is a junction between ovule and funicle. Ovule and funicle. Each ovule has one or two protective envelopes called integuments. One or two protective covering called integuments. Integuments encircle the ovule except at the tip, where a small opening called micropyle is organized. So what is the place where there are no integuments? Micropyle. Opposite the micropylar end is the chalaza, representing the basal part of the ovules. Which becomes the basal part? It is the chalaza which becomes the basal part, and micropyle becomes the apical part. Apical part. Okay. Enclosed within the integuments is a mass of cells called the nucellus. Cells of the nucellus have very important. Nucellus is very important. Abundant reserve food materials, and by the help of these food materials, what they can provide is nourishment. What they can provide is nourishment. Located in the nucellus is the embryo sac or female gametophyte. See, what is the other term for embryo sac? Female gametophyte. An ovule generally has an embryo sac formed from a megaspore. So how an ovule is generally produced? From the megaspore present inside the embryo sac. An ovule generally has a single embryo sac formed from megaspore. So an ovule generally has a single embryo sac formed from the megaspores. The process of formation of this megaspore is called as megasporogenesis. Megasporogenesis. So let us talk about this megasporogenesis. Achha, one thing you can see, this synergids, this filiform apparatus are nothing but the what? The protrusion of this synergids. Protrusion of this synergids. So megasporogenesis means the process of formation of megaspores from the megaspore mother cell. It's called megasporogenesis. In angiosperms, microsporogenesis and megasporogenesis involves meiosis. So definitely in angiosperms, microsporogenesis and microspore mother cell is going to show meiosis, and megaspore mother cell is going to show meiosis to give rise to embryo sac. Give rise to embryo sac. Okay? Now, okay. Ovules generally differentiate a single megaspore mother cell. Ovules generally differentiate a single megaspore mother cell, MMC, in the micropylar region of the nucellus. It has a large cell containing dense cytoplasm and a prominent nucleus. So, see, megaspore mother cell. Megaspore mother cell, it is a large cell containing dense cytoplasm and a prominent nucleus. I think if you see in the chapter called cell cycle and cell division, nucleus-cytoplasmic ratio, if it is increased, it is going to grow and it's going to show cell division. So meiosis is going to occur in megaspore mother cell. That's because they have got a large cell and with a prominent nucleus and a dense cytoplasm. The megaspore mother cell undergoes meiotic divisions. Meiosis results in the production of 4 megaspore. अभी हमने बताया इसको कि 4 megaspore बनाएंगे वो female gametophyte. When we talk about embryo sac, in a majority of flowering plants, one of the megaspore is functional, while the other three degenerates. Functional megaspore develops into a female gametophyte. So female gametophyte, this embryo sac बनता कहाँ से है? Functional megaspore से, not megaspore tetrad. क्योंकि 3 नष्ट हो जाएंगे, उसमें से एक functional, only one functional megaspore will be there. One functional megaspore ultimately is going to form into embryo sac. This method of embryo sac formation from a single megaspore is called monosporic development. Star mark for NEET examination. Monosporic development means what? When embryo sac is produced from a single megaspore. a single functional megaspore. Okay, the nucleus of the functional megaspore divides mitotically to form two nuclei which move to the opposite poles, forming the two nucleated embryo sac. So the nucleus of the functional megaspore, think about it, divides mitotically to form two nuclei which moves to the opposite poles, forming the two nucleated embryo, and that is called as first free nuclear division. First free nuclear division. Two more sequential mitotic nuclear divisions. That means we are calling the free nuclear divisions as what? Mitosis. So how many meiosis took place? Only one with the megaspore mother cell. And how many mitosis will take place? Three mitosis. That's three free nuclear divisions. Two more sequential mitotic nuclear divisions results in the formation of the 4 nucleate and later the 8 nucleate stage of the embryo sac. I think आपको समझ में आ गया ये, है ना? तो ये था functional megaspore. This was the functional megaspore. The first mitotic division, free nuclear division number 1. Free nuclear division number 1 results into 2 cells, 2 nucleated, then 4 nucleated, then this is the second mitosis, third mitosis. 1, 2, 3, 4, 5, 6, 7, 8. And now this is how they have formed in. Okay, there are 3 antipodal cells, polar nuclei, 1 egg cell and 2 synergies. This is how it's being formed. It is of interest to note that these mitotic divisions are strictly what? Nuclear, free nuclear. Now what is the meaning of free nuclear? Very, very important. That means only nuclear division is going to occur without cell wall formation or without cytogenesis. That is, nuclear divisions are not followed immediately by cell wall formation. After the 8 nucleated stage, the cell walls are laid down, leading to the organization of the typical female gametophyte or embryo sac. 6 of the 8 nuclei are surrounded by a cell walls, are organized into the cells. The remaining 2 nuclei, क्या कहते हैं उसको? Polar nuclei. क्या करेंगे? एक साथ रहेंगे. Are situated below the egg apparatus in the large central canal, central cell. 3 cells are grouped together at the micropylar end. क्या बनाएगा? Antipodal cells, and constitute the not. 3 cells are grouped together at the micropylar end. मतलब एक cell रहेगा और 2 synergids. Together they are called as egg. So egg cell and 2 synergids together is going to make what? Egg apparatus. The synergids have special cellular thickenings at the micropylar tip called filiform apparatus. So cellular thickenings, special cellular thickenings which play an important role in guiding the pollen tubes into the synergids. This is very, very important concept. So the filiform apparatus guides the pollen tube to enter inside the synergids. It guides the entry of the pollen tube inside the synergids. It's a very, very important concept. Now 3 cells at the chalaza. What are those called? Antipodal cells. The large central cell has 2 polar nuclei. Arrangement of nuclei in normal embryo sac is 3 plus 2 plus 3. What is 3 plus 2 plus 3? 3 means antipodal cell. This is 1 egg, 2 synergid. This is also 3. This is 3 and 2 polar nuclei. 3 plus 2 plus 3. A typical angiosperm embryo sac at maturity, though 8 nucleated and 7 celled, should not be though. For the formation of embryo sac, 1 meiotic. Yes. For the formation of embryo sac, 1 meiotic and 3 mitotic divisions are required from MMC, megaspore mother cell. This is star point about development of female gametophyte. Okay? I think that is okay. Now let us start with the new topic, and that is called embryo sac. See what happens in embryo sac. Embryo sac is to ovule as pollen grain is to anther. है ना? Embryo sac ovule के जैसा है और pollen grain is just like an anther. Starting from the innermost part, the correct sequence of parts in an ovule are egg, embryo sac, nucellus and integument. In a flower, if the megaspore mother cell forms megaspores without undergoing meiosis, and if one of the megaspores develops into an embryo sac, its nuclei would be diploid. Its nuclei would be diploid. Clear? Let's start with this. The next topic is what? Pollination. Before we get into pollination, let us describe something about pollination. We know pollination is a very, it's not for the first time you are studying pollination. So get this idea first about pollination. About pollination. What is pollination? It is a process of transfer of pollen grain from the anther to the stigma of the same or different flower of the same or different plant of the same species. That means, on the basis of pollination, we can classify them into three types. One is autogamy, next is geitonogamy, and the last one is xenogamy. So when we talk about autogamy, auto means what? Self, gamy is marriage. That means we are talking about self-pollination. This is basically self-pollination. When the pollen grains from the anther falls to the stigma of the same flower, falls to the stigma of same flower of the same flower of the same plant of the same plant of the same species. So यहाँ पे तीनों सेम है. स्पीसीज तो सेम होगा ही, सेम प्लांट भी है और फ्लावर भी सेम है. Then this is called autogamy. और autogamy होगा तो there is no chance of any variation. Fine. So nature never selects autogamy. Basically nature wants to go for variation, and that is only been done by cross pollination. Now what is geitonogamy? Geitonogamy में क्या होगा? Geitonogamy में क्या होता है? Now the transfer of pollen grain from anther to the stigma of the different flower. Very good. Of the different flower of same plant of same species. यहाँ पे देखो एक सिर्फ डिफरेंट हो गया. क्या? फ्लावर डिफरेंट हो गया. प्लांट एक ही है. यहाँ पे आकर पोलन ग्रेन यहाँ पे आकर इस स्टिग्मा पे आ गया दूसरे फ्लावर में. सो देयर विल बी नो जेनेटिक चेंज. ये जेनेटिक सिमिलर होगा. दिस इज गोइंग टू बी जेनेटिकली सिमिलर. बट देखने से कैसा लग रहा है? लग रहा है कि क्रॉस पॉलिनेशन हो रहा है, बट बेसिकली इट इज नॉट क्रॉस पॉलिनेशन. सो दिस इज वेरी इंपॉर्टेंट फॉर योर नीट एग्जामिनेशन. गीटोनोगैमी. Come down to xenogamy. When we talk about xeno means what? Foreign. Gamy is marriage. So when the transfer of pollen grain takes place from the anther to the stigma of different flower of different plant of same species. Is this clear? This is called xenogamy. This is perfect cross pollination which leads into variation. Examples of this pollination is very, very important. I think आपको समझ में आ गया ये what is pollination. Now pollination देखो क्या क्या है यहाँ पे. The male and female gametes in flowering plants are produced in the pollen grain called embryo respectively. As both types of gametes are non-motile, they have to be brought together for the fertilization. कौन लाएगा? वो तो खुद मोटाइल नहीं है, तो कोई एजेंट चाहिए. And those are called pollinating agents. It can be bees, it can be birds, it can be snail, it can be snakes, it can be human, it can be birds. So pollination is a mechanism to achieve this objective. Okay. Transfer of pollen grains shed from the anther to the stigma of the pistil is called pollination. ठीक है? अब यहाँ पे देखो pollination, autogamy, geitonogamy and xenogamy. Transfer of pollen grains from anther to the stigma of the same flower. यहाँ पे transfer pollen grains from anther to the stigma of the another flower of the same plant, or transfer pollen grains from anther to the stigma of different plant. It's called xenogamy. ठीक है? Okay. Next. Flowering plants have evolved an amazing array of adaptations to achieve pollination. They make use of external agents to achieve pollination. Fine. Flowering plants have evolved an amazing array of adaptations. क्या adaptation लगता है आपको? What are the adaptations they can have to attract the pollinators? Have vibrant colors, sometimes smell, nectar which acts as a floral reward, है ना? तो ये सब उसमें adaptations हैं plant में. Kinds of pollination, depending on the source of the pollen, pollination can be divided into three types. One is autogamy. In this type of pollination, achieved within the same flower, transfer pollen grains from anther to the stigma of the same flower. In a normal flower which opens and exposes the anthers and stigma, complete autogamy is rather rare. Autogamy in such flowers requires synchrony in pollen release and stigma receptivity. ये थोड़ा आपको समझना है. Autogamy in such flowers requires synchrony, that is synchronization, timing सेम होना चाहिए. क्या किसका? Pollen release का and stigma का receptivity का. अगर ऐसा होता है पोलन तो रिलीज हो गया पर स्टिग्मा रेडी नहीं है, तो इट्स नॉट गुड आपका. फाइन. ऑल्सो द एंथर्स एंड द स्टिग्मा शुड लाई क्लोज टू ईच अदर सो दैट सेल पॉलिनेशन कैन ऑकर. ठीक है, तो उनमें क्या होना है, पास पास आना है ताकि सेल पॉलिनेशन हो. इन सम प्लांट्स सच एज वायोला, कॉमन पैंसी, वेरी वेरी इंपॉर्टेंट, द एग्जांपल्स आर वेरी इंपॉर्टेंट. ऑक्सैलिस एंड कामेलिना प्रोड्यूस टू टाइप्स ऑफ फ्लावर्स, chasmogamous, which are similar to open flowers of other species with exposed anthers and stigma, and cleistogamous. Cleistogamous मतलब closed flowers. Chasmogamous मतलब क्या है, open flower, which do not open at all. तो जब ये open नहीं करेगा cleistogamous, तो उसमें क्या होने वाला है, self pollination is the only possibility. Self pollination is the only possibility in case of cleistogamous flower, but in case of chasmogamous flower, the open flower, तो यहाँ पे क्या होने वाला है, सेल्फ पॉलिनेशन भी हो सकता है, क्रॉस पॉलिनेशन भी हो सकता है. इन सच फ्लावर्स द एंथर्स एंड स्टिग्मा लाई क्लोज टू ईच अदर. व्हेन एंथर dehisce in the flower bud, pollen grains come in contact with stigma to effect pollination. फाइन. देखो यहाँ पे क्लिस्टोगैमस दिया है, दे डू नॉट ओपन, बंद है पूरा फ्लावर, और ये ओपन फ्लावर है, मतलब यहाँ पे क्या होगा, बाहर से भी आ सकता है और खुद में भी हो सकता है चैसमोगैमस फ्लावर. Thistous flowers are invariably autogamous, है ना, invariably autogamous, कोई चांस नहीं है क्या होने का, xenogamy होने का, as there is no chance of cross pollination landing on the stigma. Cleistogamous flowers are produce assured seed set even in absence of pollinators. उसको तो pollinator चाहिए नहीं, तो सीड का सेट उनका 100% सेम रहेगा. फाइन, वेरी गुड. Cleistogamous, transfer pollen grains from anther to the stigma of another flower of the same plant. सेम प्लांट होगा पर डिफरेंट फ्लावर में होने वाला है. ऑल्दो गिटोगमी इज फंक्शनली क्रॉस पॉलिनेशन, देखने में लगता है क्रॉस पॉलिनेशन हो रहा है, इन्वॉल्विंग अ पॉलिनेटिंग एजेंट जेनेटिकली सिमिलर, वेरी वेरी इंपॉर्टेंट. सो गिटोगमी में क्या होगा, इट इज गोइंग टू फॉर्म इनटू जेनेटिकली सिमिलर टू ऑटोगमी. क्लियर? सिंस द पोलन ग्रेन्स कम्स फ्रॉम द सेम प्लांट से आ रहा है, इसलिए जेनेटिकली सिमिलर होगा. व्हेन वी टॉक अबाउट xenogamy और क्रॉस पॉलिनेशन, ट्रांसफर ऑफ पोलन ग्रेन्स from anther to the stigma of the different plant, this is the only type of pollination which during pollination brings genetically different types of pollen grains to the stigma. तो यही है जो असल में क्रॉस पॉलिनेशन है. दिस इज बेसिकली द क्रॉस पॉलिनेशन ऑफ इट. क्लियर? ओके, नेक्स्ट, outbreeding device. Now what is the meaning of outbreeding device? Now understand, nature wants what? Cross pollination or self pollination? Nature wants cross pollination to occur because the nature wants variation should be there. Now variation, nature cross pollination के लिए क्या क्या promote करते हैं plant में? What are the structural changes that a plant should have in order to confirm cross pollination? Number one, और उसको क्या कहते हैं, outbreeding device. Outbreeding, not inbreeding. Inbreeding से क्या होगा, अगर इनब्रीडिंग होगा बार बार तो क्या होगा, इनब्रीडिंग डिप्रेशन हो जाएगा, डिप्रेशन हो जाएगा. सो आउटब्रीडिंग चाहिए, क्रॉस पॉलिनेशन चाहिए. सो मेनी ऑफ फ्लावरिंग प्लांट्स प्रोड्यूस hermaphrodite flowers, that is bisexual flowers, and pollen grains are likely to come in contact with the stigma of the same flower. Continued self pollination, अगर बार बार सेपरेशन होगा, results in inbreeding, important, inbreeding depression. Flowering plants have developed many devices to discourage self-pollination and encourage cross-pollination. First device क्या है, in some species pollen release and stigma receptivity दोनों में क्या होगा, do not synchronized, are not synchronized. इसको क्या कहते हैं, what is the term for that? It is called as dichogamy. क्या कहते हैं इसको, ये याद रखना है, बुक में ये टर्म नहीं दिया गया है, पर अगर आ जाता है तो आपको याद रखना है. So what is the term? Dichogamy. Dichogamy are of two types. One is called protandry, और एक है प्रोटोगाइनी. एंड्री मतलब क्या है, एंड्रोशियम. प्रोटो मतलब फर्स्ट, तो तो अगर एंड्रोशियम पहले बड़ा हो गया, मेल रिप्रोडक्टिव ऑर्गन पहले बड़ा हो गया, प्लांट है बाइसेक्सुअल, फ्लावर है बाइसेक्सुअल, एंथर बड़ा हो गया, स्टिग्मा अभी भी इमैच्योर्ड है, मतलब स्टिग्मा का रिसेप्टिविटी इज नॉट सिंक्रोनाइज्ड। तो ये पोलन ग्रेन अगर स्टिग्मा में भी गिरेगा भी तब भी क्या नहीं होगा, फर्टिलाइजेशन नहीं होगा। तो ये एक सिंक्रोनाइजेशन का चेंजेस से, डाइकोगैमी से क्रॉस पॉलिनेशन इज ऑलवेज पॉसिबल, सेल्फ पॉलिनेशन is avoided. So the term is dichogamy, protantry and protogamy. Either the pollen is released before the stigma becomes receptive, or stigma becomes receptive much before the release of pollen. Second device. In some species, the anther and stigma are placed at different positions so that the pollen cannot come in contact with the stigma. पोजिशंस डिफरेंस है, बहुत लंबा एंथर है, छोटा है स्टिग्मा, या स्टिग्मा बहुत लंबा है, छोटा है एंथर। सो इन दैट केस इसका हेटेरोस्टाइली, क्या कहते हैं इसको, हेटेरोस्टाइली। इट्स गोइंग टू प्रमोट व्हाट, इट्स गोइंग टू प्रमोट क्रॉस पॉलिनेशन, नॉट सेल्फ पॉलिनेशन। बोथ दिस डिवाइसेस आर टू प्रिवेंट ऑटोगैमी। ये दोनों डिवाइस, फर्स्ट और सेकेंड, दैट इज डाइकोगैमी एंड हेटेरोस्टाइली, ये दोनों किसमें प्रिवेंट कर रहे हैं, ऑटोगैमी को प्रिवेंट कर रहे हैं। थर्ड डिवाइस क्या है, थर्ड डिवाइस है सेल्फ इनकम्पैटिबिलिटी। इनब्रीडिंग को बंद करने के लिए खुद में सेल्फ इनकम्पैटिबल है प्लांट्स, और सेल्फ स्टेरिलिटी कहते हैं इसको, सेल्फ स्टेरिलिटी। दिस इज अ जेनेटिक मैकेनिज्म एंड प्रिवेंट्स सेल्फ पोलन फ्रॉम द सेम फ्लावर और अदर फ्लावर्स ऑफ द सेम प्लांट फ्रॉम फर्टिलाइजिंग द ओव्यूल्स बाय इनहिबिटिंग पोलन जर्मिनेशन और पोलन ट्यूब ग्रोथ इन द पिस्टिल। ठीक है, तो ये स्टिग्मा है, सेल्फ पोलन ग्रेन आकर गिरा यहाँ पे, तो ये इनकम्पैटिबल है, इससे पोलन ट्यूब का ग्रोथ ही नहीं करने देगा। ये सेल्फ इनकम्पैटिबिलिटी और सेल्फ स्टेरिलिटी। पर अगर बाहर से कोई पोलन ग्रेन आए, इफ फ्रॉम आउटसाइड अ पोलन ग्रेन फॉल्स ओवर हियर, देन इट इज गोइंग टू स्टार्ट ग्रोइंग द पोलन ट्यूब towards the megasporangium or ovule. Fourth device, to prevent cell pollination, plant produces unisexual flowers, diclining or unisexuality. ठीक है, सेल पॉलिनेशन को बंद करने के लिए अगर यूनिसेक्सुअल फ्लावर बन गया, नॉट बाइसेक्सुअल, तो ऑलवेज देयर इज अ चांस ऑफ व्हाट, क्रॉस पॉलिनेशन। If both male and female flowers are present on the same plant, such as castor and maize, that is monoecious. Example important है, मेल एंड फीमेल फ्लावर दोनों एक ही प्लांट में प्रेजेंट है, वी कॉल इट मोनोशियस, दैट मींस दे आर बाइसेक्सुअल। इट प्रिवेंट्स ऑटोगैमी बट नॉट गिटोनोगैमी। वो ऑटोगैमी को बंद कर सकता है पर गिटोनोगैमी को बंद नहीं कर सकता है। सो कैस्टर एंड मेज। In several species such as papaya, male and female flowers are present on different plants. That is, each plant is either male or female, called dioici. Dioici मतलब dioecious, unisexual. So papaya is the best example of unisexual flowers. तो उसमें क्या होने वाला है? 100% cross pollination. This condition prevents both autogamy and geitonogamy. देखो NEET 2017 में आ गया। Dioecious plant bearing only male or female flowers presents both autogamy and geitonogamy. Cleistomas flower prevents both geitonogamy and xenogamy. है ना? Cleistomas मतलब बंद फ्लावर, क्लोज फ्लावर, वो क्या प्रिवेंट करेगा? geitonogamy and xenogamy both it is going to prevent. Agents of pollination. What are the agents of pollination? Agents of pollination can be abiotic और बायोटिक। बायोटिक में एनिमल होगा, इंसेक्ट होगा, एमोर्फोफैलस, यक्का, फिग, याद है? अबायोटिक प्लांट्स में, अबायोटिक एजेंट्स में होगा विंड, मेज, ग्रासेस, वाटर, हाइड्रिला, वैलिसिडिया, जॉस्टेरा। एग्जांपल्स इंपॉर्टेंट है, बहुत इंपॉर्टेंट है। एग्जांपल्स आर मोस्ट इंपॉर्टेंट। Plants use two abiotic, wind and water, and one biotic agents. Majority of plants use biotic agents for pollination. Only a small proportion of plants use abiotic. Pollen grains coming in contact with the stigma is chance factor in both wind and water. To compensate for these uncertainties and associated loss of pollen grains, the flowers produce enormous amount of pollen when compared to the number of ovules available for population. है ना? Pollination के लिए फ्लावर क्या ज़्यादा बनाते हैं? पोलन ग्रेन ज़्यादा बनाते हैं to assure कि एक पोलन ग्रेन तो जाकर पहुंचेगा। Now, name the particular animal which constitutes 96% of the pollination in the world. Is a bees, social insect bees. Okay. Anemophily. So wind pollination. Wind pollination क्या कहते हैं? Anemophily. So before we get into the pollinating agents, let's get some of the pollinating agents examples. Pollinating agents examples. Pollinating agents examples. Okay. Types. It can be wind. When it is called wind, we call it anemophily. We call it anemophily. Second, if it is water, we call it hydrophily. We call it hydrophily. Third, if it is insect, we call it entomophily. It's a highly important term for NEET. Entomophily is highly important point for NEET, insects. If it is ants, it is called as myrmecophily. Myrmecophily. If it is bird, we call it as ornithophily. Ornithophily. Tell me. If it is snail, we call it as malacophily. If it is snakes, if the pollination is by the help of snakes, ophiophily. Ophiophily. If it is by the help of human, you call it anthropophily. Anthropophily. If it is by the help of bats, you call it chiropterophily. Pterophily. If it is by the help of elephants, it is called as elephophily. Elephophily or lophophily. So these are the different types of agents. Fine. The most important which is there in your book are insects. Wait. Okay. So there should be some adaptations of the plants to attract insects. तो वो देखना है. और wind pollination है तो क्या होगा? Sticky होगा, scattered होगा. So we will talk about that and those adaptations also. Okay, let's start with anemophily, wind pollination. Pollination by wind is more common amongst abiotic pollination. सबसे ज़्यादा होगा abiotic pollination में this way, wind से. Anemophilous flowers make certain adaptations. ये important है. क्या है? Flowers are small and inconspicuous. Non-scented, scent नहीं होगा, without bright colors, nectar and fragrance. Wind pollination also requires that the pollen grains are light and non-sticky. Pollen grain को light होना है, non-sticky होना है, wind currents. They often possess well-exposed stamens. Stamens well-exposed होना चाहिए, well-exposed stamens. Pollens are easily dispersed into the wind currents. And large feathery stigma. Stigma should be feathery to trap airborne pollen grains. Wind pollinated flowers often have a single ovule in each ovary. ये important है. Wind pollinated flowers are often having a single ovule in each ovary and numerous flowers packed into inflorescence. Clear? So that is about wind pollination. The examples. Wind pollination is quite common in grasses, Camelina, Amaranthus. Okay, familiar example is the corn cob. Okay, very important. Corn cob, tassels, the tassels, very important. Corn cob, I think you have seen this corn cob. They are hairy, beard-like structures. You have seen corn cob. You see nothing but the stigma and the style which wave in the wind to trap. Hydrophily. Hydrophily, water pollination. Pollination by water is quite rare in flowering plants and is limited to about 30 genera, mostly monocotyledons. As against this, water is a regular mode of transport for the male gametes among the lower plant groups such as algae, bryophytes and pteridophytes. It is believed, particularly for some bryophytes and pteridophytes, that their distribution is limited because of the need of water for the transfer of male gametes and fertilization. Some examples of water-pollinated plants are Vallisneria hybrida. ठीक है, याद रखना पड़ेगा आपको. With growing fresh water and several marine sea grasses such as Zostera. So Zostera is a marine seagrass. Zostera. Not all aquatic plants use water for pollination. In a majority of aquatic plants such as water hyacinth and water lily, the flowers emerge above the level. So though water hyacinth and water lily are aquatic plants, but their flowers they emerge above the water body. So they help in what? They go for wind pollination, mind you, not water pollination. Okay, so that's it about hydrophily. I think in Vallisneria, the female flowers reach the surface. The female flowers reach the surface of the water by the long stalk, and the male flowers or pollen grains are released onto the surface of the water. They are carried passively by the water. Passively, automatically by the water currents. Some of them eventually reach the female flowers on the stigma. In another group of water-pollinated plants such as sea grasses, is Zostera. Female flowers remain submerged in water. Female flowers कहाँ पे रहते हैं? They remain submerged in the water, and the pollen grains are released inside the water. Pollen grains in many such species are long ribbon-like. Very, very important. Star mark. Pollen grains in many such species are long ribbon-like, and they are carried passively inside the water. Some of them reach the stigma and achieve pollination. Most of the water-pollinated species, pollen grains are protected from wetting by a mucilage covering. Very, very important. Mucilage covering. So mucus is there which prevents the pollen grains from being affected by fungus. Both wind and water pollinated flowers are not very colorful and do not produce nectar. Not required nectar. किसको बुलाएगा? Nectar is going to attract insects. So wind and water both are abiotic, so they don't need any life. Come now to zoophily. That is pollination by the help of animals. Majority of the flowering plants use a range of animals such as pollinating agents. Bees, butterflies, flies, beetles, wasps, ants, moths, birds, sunbirds and hummingbirds and bats are the common pollinating agents. Even larger animals such as some primates, lemurs, arboreal tree dwellers, rodents or even reptiles, gecko lizard and garden lizard have also been reported as pollinators in some species. Among the animals, insects, particularly bees, are the dominant biotic pollinating agents. Often flower of animal pollinated plants are specially adapted for a particular species of animals. So when we talk about zoophily, I think we have talked about wasps and fig. So fig is normally, it's an example of mutualism. Fig is normally pollinated by wasps. And I think we have talked about beetles also. Bumblebee, they normally, they reach the Ophrys, Mediterranean Ophrys, that is orchid plant, and copulates with one of the petals which looks similar like a female bumblebee. So there is also a way that they can help in what? Pollination. So zoophily is done. Majority of the insect pollinated flowers are large, colorful. So they should be colorful because they should get a floral reward. Large, colorful and fragrant and rich in nectar. Example, lotus. When the flowers are small, a number of flowers are clustered into an inflorescence to make it conspicuous. Animals are attracted to flowers by color or fragrance. The flowers pollinated by flies and beetles create foul odors to attract these animals. To sustain animal visits, the flowers have to provide rewards to the animals. These are called floral rewards in the form of nectar, and pollen grains are usually floral rewards. For harvesting, the reward from the flower, the animal visitor comes in contact with the anthers and stigma. The body of animals gets a coating of pollen grains, which are generally sticky in animals. So they don't know that they have got pollen grains on their body and help in pollination. When the animal carrying pollens on its body comes in contact with the stigma, it brings about pollination. Attractants and rewards are required for entomophily. Attractants and rewards are required for entomophily, that is insect pollination. In some species, Floral dewards are in providing safe places to lay eggs. Come on, tell me the example. Oviposition. Some of the moths, Pronuba moth, they reach the yucca plant, fine, and what they do is they seek the locule of the ovary where they are going to lay eggs, and we call this as oviposition. So the tallest flower of Amorphophallus, the flower itself is about 6 feet in height. So it is pointed by a moth. What is written? Okay, a similar relationship exists between a species of moth. What is the name of the moth? Pronuba. And the plant yucca, where both species, moth and the plant, cannot complete their life cycle without each other. So cannot complete their life cycle without each other. What kind of relationship they have? Obligatory or non-obligatory? Obligatory. Strict relationship है. एक दूसरे के बिना मर जाएगा. So this is called obligatory relationship, which we call it as mutualism. Non-obligatory होता तो क्या कहते हैं उसको? Mutualism नहीं कहते, उसको protocooperation कहते हैं. The moth deposits its eggs in the locule of the ovary, and the flower in turn gets pollinated by the moth. The larvae of the moth come out of the eggs as the seeds start developing. Many insects may consume pollen or the nectar without bringing about pollination. Such floral visitors are referred to as pollen robbers. Pollen वो क्या करते हैं? पोलन ग्रेन को कंज्यूम कर लेते हैं, खा लेते हैं, एंड नेक्टर को भी खा लेते हैं, पर क्या नहीं करते हैं? नो पॉलिनेशन इज डन बाय देम, सो दे आर पोलन रॉबर्स और नेक्टर रॉबर्स. अच्छा नाम दिया है ना. एनिमल वेक्टर्स आर रिक्वायर्ड फॉर पॉलिनेशन इन कुकुम्बर. एनिमल वेक्टर्स आर रिक्वायर्ड फॉर पॉलिनेशन इन कुकुम्बर. NEET 2013, huh? Very important. Pollen pistil interaction. Pollen pistil interaction. Pollen pistil interaction, I think I'm going to start in the next class. Let us solve some questions. I think let us complete it. It's only there. Pollen pistil interaction. So what is the meaning of this? Interaction should be there. Just like in human beings, a human sperm reaches towards the ovum. There is a chemical dialogue. The ovum secretes fertilizing. The human sperm releases anti-fertilizing. So there is a chemical dialogue by which the sperm can reach up to the ovum. Just like that, in case of plant also, there should be pollen pistil interaction. Fine. Often pollen of the wrong type, either from other species or from the same plant, if it is incompatible, also land on the stigma. The pistil has the ability to recognize the pollen. Pistil में ability है किसको recognize करने के लिए? Right type of pollen है कि नहीं? Fine. Whether it is of the right type, compatible, or the wrong type, incompatible. If the pollen is of the wrong type, the pistil rejects the pollen by preventing pollen germination or on the stigma of the pollen tube growth in the style. Growth होने नहीं देगा. The ability of the pistil to recognize the pollen followed by its acceptance or rejection is the result of a continuous dialogue between pollen grain and the pistil. This dialogue is mediated by a chemical components of the pollen interacting with those of the pistil. Pollen grains germinates on the stigma to produce a pollen tube through one of the germ pores. है ना? Germ pore जानते हैं नहीं जानते हैं? Yes, I think we know germ pore. If this is a pollen grain, this is exine, and this is germ pore. So exine is this is intine. The place where exine meets intine is called germ pore. Sorry. The contents of the pollen grains move into the pollen tube. Okay? Okay. In some plants, pollen grains are shed at two cell stage as vegetative cell and generative cell. In such plants, the generative cell divided and forms the two male gametes during the growth of the tube in the stigma. We have talked about that. Pollen grain we have already discussed. 2 cell and 3 cell stage. In plants which shed pollen in the 3 cell stage condition, pollen tubes carry the two male gametes from the beginning. Transmission tissue is the characteristics of solid style. Pollen tube, after reaching the ovary, enters the ovule through micropyle and then enters on one of the synergids through the filiform apparatus. Filiform apparatus presents at the micropyle part of the synergids, guides the entry of the pollen tube. That is very, very important. We have talked about that. All these events from pollen deposition on the stigma until pollen tubes enter the ovule are together referred to as pollen pistil interaction. To study pollen germination, the pollen from pea, chickpea, crotalaria, balsam and vinca containing a drop of sugar solution about 10%. After about 15 to 30%, 30 minutes, pollen tube coming out of the pollen grains. That's all about pollen pistil interaction. Let us start some of the questions. Do the first one, the hilum. Option D. Seed where the funicle was attached. Which of the following are the important flow rewards? Important floral rewards to the animal pollinators. Nectar and pollen grain, floral fragments and calcium crystals, protein and follicle, color and last size. A. Phylum apparatus is the characteristic feature of synergids. Geitonogamy involves fertilization of flower by the pollen tube, pollen from another flower of the
same plant.
another flower of the same plant. A is the answer. Male gametophyte. Least number of cells. No idea, I think. Lilium. The answer is lilium. Male gametophyte with least number of cells is lilium. Function of filiform apparatus is to
guide the entry of pollen.
Guide the entry of pollen tube. Very easy. Yes. Which one of the following
Is correct.
is correct?
C.
Okay. Which of the following statement is correct?
B.
Chasma flowers do not open at all. Chasma flowers do not open at all. They are open. Is correct.
Option B.
Chrysanthemum flowers are always autonomous. Megaspores are produced from the
After the spores,
mutic division. Mutic division. That's all, I think. That's all about today's class. We have yet to complete it. This is a huge topic. So next day we are going to talk about the rest of it, and we are going to complete the chapter in the next very class. Okay, start solving questions. Fine, question solve करो जितना करो. याद रहे 12,000 क्वेश्चंस करने के बाद अगर आप नीट में बैठते हो 180 क्वेश्चंस आपको कौन मिलेगा, एंड दैट इज फॉर श्योर. फाइन. और एक दिन में 12,000 क्वेश्चन नहीं होता है, सो एक स्टेप से होता है. सो स्टार्ट डूइंग दैट. ओके, ऑल द बेस्ट.